Okay, good afternoon to all of you. Welcome in this video lecture. In this chapter, that is amino acid and protein, half of the portion, that is the amino acid, we have completed, and from this video lecture we are going to continue the second half, that is the proteins. The lecture includes general information of protein, definition of protein, and some of the types of protein according to their responsibilities. Now, wherever the protein as a biomolecule is considered, no doubt proteins are the macro large molecules made up of from monomers called as amino acids. That is, we already studied. So, amino acids are the building blocks of all proteins, and proteins are naturally developed by using various alpha amino acid as a key precursor. So, proteins are simply defined as the macrobiomolecules which are developed from monomers, that is alpha amino acids, and plays an important role in various biological processes. This is a very simple definition. See the slide. Whenever the various kinds of living organisms, human body, animal body is considered, then in the muscle there are various kinds of muscles, bones, skin, hair, eyes. Such type of various parts of the body are composed from different tissues. These all tissues are proteinous material. Yes. So our body is containing proteinous material in the form of muscle, bone, skin, hair, and virtually every living part of the body made up of from protein, and they are made up of from 20 different types of alpha amino acids. How many? That is 20. Now they are structurally arranged in a various types, and by the combination of 20 different alpha amino acid we are getting around 10,000 proteins. Even this quantity is more. So this is very important. That is near about 10,000 and above number of proteins are developed from 20 alpha amino acids. So this is simply definition and use of that particular proteins. Now whenever the various key properties of proteins are considered which are responsible for their functions, first important key property is the protein's structural form. What is the structure of protein? Structural forms of protein. No doubt, proteins are constructed linearly from 20 alpha amino acids, which are recurring process in the cell, means it is a regular process in the body, living cells. But whenever linear or primary sequence of protein is considered, they are responsible for their functional role. Means as the protein is developed from alpha amino acid, it gives the development of new protein and having specific functional role. So the sequence spontaneously fold into different arrangement, forming three-dimensional structure and depend on this series of combination of alpha amino acid, the various proteins are developed and they decide which type of function is completed by protein. So structural form plays an important role. This is protein structural form. Second, proteins with different functional groups. Proteins with different functional groups. This is the second key property. No doubt, most of the predominating proteins are composed by the combination of various kinds of alpha amino acid and they interact with other proteins. That is, particularly various proteins are developed from alpha amino acids and their functional group of protein are alcoholic OH group, thiols, thioethers, carboxylic acid, carboxamide, and variety of basic groups. So most of the proteins are having functional group like alcoholic OH group, thiolic OH group, SH group means, then thioethers, then carboxylic acid COH, then carboxamides, and such kind of basic groups are present in the protein as a functional group. So these functional groups are randomly combined during the development of protein and gives formation of sequence of alpha amino acids. So according to this, the functionality of protein is also depends on which kind of functional group is present in that particular protein, and this protein is now further acting extension of protein is the enzymes and hormones, its biomolecules. So functional groups present in the protein are carboxylic acid functional group, thiols, thioethers, carboxamides. So these are the various functional groups present in the protein. So this is second key property. Now third is interactions of protein forming complex assemblies. See the point? No doubt protein are the large biomolecules, macromolecules, and they play an important role in our day-to-day metabolism. No doubt we are taking protein from various types of food material, right? So this food material contains various proteins and these proteins are so many interaction with other proteins, alpha amino acids, so that they are giving a various complex assemblies. For example, DNA replication. Now see, DNA is composed from various kinds of protein and all of we are much more familiar that each and every living moiety body is having various types of protein. Human body is also having its own DNA, each and every. So this intracellular transmission of signal and complex essential biological process is carried out by interactions of proteins. So this is very important biological process which is given by the proteins. So this is the third key property, and fourth important is rigidity and flexibility of protein. Rigidity. Rigid protein means what? The protein structure which is fixed and compact, which restrict any motion or movement. Such type of protein called as rigid protein. Such type of protein is called as rigid protein. That is protein which is having a restriction to movement and motion. Such kind of protein is called as rigid protein. And second kind of protein is they are a rigid type of protein is particularly involved to develop structural formation of cell. What is the use of rigid protein? That is, rigid proteins are used to form a structure of different cells. which is fixed, not changeable. For example, cytoskeleton, which forms internal scaffolding within a cell. In the cell, fixed skeleton, that is cytoskeleton, is developed by rigid type of protein. It gives formation of rigid type of protein. And now second type of protein is the flexible protein. Now what is the role of flexible protein? Flexible protein is used in the cell, which is loosely bounded in structure, loose, not firm, and it can be easily rearranged or moved when required. So the role of flexible protein is it is loosely attached to fixed skeleton and easily move from one biological site to another biological site. It is simply act as a hinges or springs or levers. So these flexible proteins are used hinges, springs or levers. They are flexible proteins and perform the essential functions of various proteins in the body. So remember these two important points. Flexible protein, which is attached loosely in the cell, they can move anywhere and rearranged for various kinds of functions. So these are the four key properties which are shown, that is structural form, functional group present in the protein, then complex assemblies and rigid and flexible proteins. Now we move towards synthesis of proteins, how the proteins are synthesized. Now see, proteins are synthesized by two manners. One is the natural or biological synthesis and another one is the chemical synthesis of protein. That is second type. Chemical synthesis means proteins are synthesized by using these two methods, biological synthesis and chemical synthesis. Now in biological synthesis, very important theme is that, see the diagram. Now in this structure of protein synthesis, you are observing different letters in the structure, starting from C, G, U, again G, A, C, A, again G, U, C, G, U, A, C, G, U, C, A, G, A, C, U. This is similar to coding, and each and every letter in this diagram in the form of C. C stands for cytosine as an amino acid, sorry, as a nucleotide. T is the thymine, G is the guanine, and A is standing for adenine. Adenine, guanine, cytosine, and thymine. All these are the four nucleotides which are present in the protein, So we first see how protein is biologically synthesized in details. Now see, the process of protein synthesis is called as translation. कशाला म्हणतो आपण translation? Translation is nothing but it is the process of protein synthesis. In this process, what happens? In this process, metabolic RNA code are translated into respective amino acids. RNA codes are translated into respective amino acids that are involved in protein formation. Means RNA which is present in any living cell or living body, it is translated into corresponding amino acids. and these amino acids are involved in protein formation. Now, each amino acid has its unique nucleotide sequence of gene. Living cell मध्ये काय असतो gene. In the living cell there is a presence of gene. Gene is composed from nucleotides, and each nucleotide is having a series of alpha amino acid. There are 20 types of alpha amino acid, and these alpha amino acid which are present in nucleotides, they are decomposed into alpha amino acid, and these alpha amino acids are used for the synthesis, biological synthesis of various protein. Now see, this is the diagram which is showing protein synthesis. See, polypeptide bond which is developed between two successive alpha amino acids. In the diagram, see the diagram, each and every colored ball is shown, sphere is shown, and each colored sphere shows different alpha amino acid. These near about 6 different alpha amino acids are combined with which bonding? Polypeptide bonding, and this polypeptide bonding gives formation of various kinds of protein. Now see, these proteins are developed from alpha amino acid, and in the living cell these alpha amino acids are released, translated from nucleotides. See, in the diagram various nucleotides are shown. In the diagram various nucleotides are shown. Ribosome दाखवलेला आहे. They have shown ribosome. RNA is composed from series of 4 nucleotides. I already discussed. That is A for adenine, then G for, that is particularly guanine, C for cytosine, and T for thymine. From nucleotides we are getting amino acids, and from amino acids by formation of peptide bond, naturally various kinds of proteins are synthesized. So this is the illustration of protein synthesis from nucleotide to amino acid and amino acid to protein, isn't it? Okay, I hope that you got this point. This is the biological synthesis of protein. Now chemical synthesis, focus on chemical method in the laboratory. Now in chemical synthesis, proteins are synthesized by formation of peptide bond. That is, chemical synthesis of protein involves peptide bond formation known as peptide synthesis. Means chemical agitation, then standard legitation, all such kinds of alpha amino acids are combined, and near about 30 to 50 alpha amino acids are combined in the laboratory by formation of peptide bond, that is C double bond O NH bond, peptide bond, polypeptides are developed, and these polypeptides are gives formation of various kinds of protein. But nowadays this technique is having some limitation. No doubt polypeptides are developed in laboratory, but chain of more than 300 alpha amino acids are not developed easily. Only limitation is that in the laboratory we can develop 30 to 40 alpha amino acids combination to give various kinds of protein. So this is simply chemical synthesis of protein from various kinds of alpha amino acid. So in this lecture we have discussed definition of protein, key property of protein, that is protein structural form, protein functional form, interaction of protein, rigidity and flexible proteins, and then important one, synthesis of protein by biological synthesis and chemical synthesis of protein. So okay, thank you. Study this particular introductory lecture. Okay, thank you. We'll stop.